I'd like to thank Dr. Latham and the RDSP committee for inviting me to share some of our work today, which is on stress and its impact on gastrointestinal diseases and how we've been using a dual model that has relevance to both human biomedical aspects as well as agricultural species. So today I'm going to cover, talk a little bit about pigs as models of gastrointestinal disease. And I will specifically point out some comparative anatomy and developmental and functional aspects of the porcine gastrointestinal tract in relation to the human. That's a little bit different from the rodent, and why this may be beneficial to studying certain types of gastrointestinal diseases. Then I will share some aspects of the research that we've been working on in the pig, which is trying to understand how early life stressors or adversity leads to GI disease susceptibility, which is a problem in both humans and animals. And then I'll cover some aspects of translational implications in the future for models of pigs in GI research. In general, the pig has a very similar gastrointestinal tract compared to the human. They are omnivores, and as mentioned before, other aspects, general aspects such as the similar size and anatomy in general for their GI tract. But specifically, there's some aspects of the porcine gastrointestinal tract that make them good models for certain gastrointestinal diseases in humans. And one of them is starting from the esophagus here, is the anatomy and structure of their esophageal epithelium, and its comparability with the stomach, with the pig, between the humans, because especially in the area of the gastroesophageal junction is more similar in the pig than it is with to humans than it is with rodents. And this is predominantly through that pigs have expression of these submucosal glands, and the transition of the esophageal epithelium into the stomach and the glandular epithelium is very similar in the pig between the human, which is not so much in the rodent, which has half of their gastrointestinal tract is predominantly non-glandular. So this makes this a great model for studying diseases such as gastroesophageal reflux disease or Barrett's esophagus and adenocarcinomas that may develop due to reflux. Another aspect of GI anatomy is the enteric nervous system, and we know that the ENS disorders are major pathophysiology of some of the most prevalent GI disorders in humans, including irritable bowel syndrome, as well as a lot of other motility disorders. And in the pig has a very complex enteric nervous system similar to the human. There's a lot of interrelated neuron projections and co-localization of neurochemicals to certain types of neurons that are not present in the rodent enteric nervous system. So this becomes important when trying to assess different potential therapies and treatments that target the enteric nervous system. The complexity of that system will be important from translating that as well. As also mentioned before, although pigs have some different aspects of their immune system that make them different from humans, in general, their immune system and their immune responses are quite similar in response to certain challenges and inflammatory conditions. And also clinical presentation of gastrointestinal disease. For example, the presentation of diarrhea in animals, which is a very common presentation in larger animal species, but to induce a clinical diarrhea in rodents is often difficult, often requiring maybe addition of chemical colitis models or severe genetic mutations to actually create a situation where there's a diarrhea, in addition to constipation. This is where pigs do develop clinical presentations of disease, which may be important when you're assessing outcomes of diseases or as well as therapies. Having a model to begin to study something that looks very similar. When talking about some gastrointestinal diseases, particularly early in life, it's important to consider some developmental differences that may exist between all the models that we use. In the gastrointestinal tract, we know that there is an extensive amount of development in many systems postnatally in the GI tract. And if you look at some specific aspects, one of them is intestinal barrier development. And the pig and the infant at term are born with a relatively well-developed gastrointestinal barrier that undergoes some maturation over the first couple of days of life, but it remains relatively stable from there. Whereas in rodents, particularly, they are born with a very immature gastrointestinal barrier, and it takes up to 2 to 3 weeks for them to actually establish this intestinal barrier function. So this becomes important when you're looking at models of early life or early damage to the gut. Another histological aspect that is different between pigs, mice, and humans is the epithelial structure. Again, pigs and humans are born with a much more developed architecture of their gastrointestinal tract, including the crypts, which are the cells that are going to proliferate and generate the epithelial cells that are lining the villus. Whereas in rodents, they are born predominantly devoid of intestinal crypt architecture, and this doesn't develop until weeks after life. So again, when you're looking at injury or early life trauma, such as diseases, whether you're looking at infections, neonatal infections, or more human-related diseases like necrotizing enterocolitis, when there's a tremendous amount of injury to the gut and there's regeneration of the barrier and the epithelium, this becomes an important consideration in the model that you're using. It was also mentioned this morning about the brain, the complexity and the similarities between the pig and the human as far as the growth and size of their brain as they developed. And I bring this into a gastrointestinal lecture because studying the complexities of the brain-gut axis and how this communicates is very important, particularly in stress-induced GI disorders, which require a complex integration of a stress response that is integrating with the peripheral systems like the gut. So a well-developed gastrointestinal system and central nervous system is critical to understanding these complex diseases. There are a number of gastrointestinal disease models out there that have been published, and this is just a list of them. I'm sure I left some out, but these can range from looking at enteric infection models, potentially as models of the piglet to study diarrheal disease in the infant, such as rotavirus. I mentioned gastroesophageal injury diseases. They make a great model for infant formula, gut development, and brain development. There's been a lot of work on the models of ischemic reperfusion, metabolic disorders, models of necrotizing enterocolitis, and some of the models that we've been working with on stress-related gastrointestinal disorders, which I'll go into more this afternoon. We study the impact of stress on the gastrointestinal tract because stress is a major predisposing factor for a number of very important and burdensome GI disorders in both animals, but as well as people as well. And one area that we are interested in is in the early life stress. So in our laboratory, we use multiple models, including the pig and the mice, to really begin to study basic mechanisms of stress-induced physiology that have applications to animals, as well as looking at whether the pathophysiology that we're seeing have implications for human diseases as well. And we utilize a number of different models. We are looking at intestinal epithelial transport, immune development, and models in the pig and the mouse, and focused on neuroimmunology in the gut and how this is impacted in development. We are particularly interested in stressors that are occurring early in life. So the early life origins of diseases that has been mentioned this morning. We know that early life adversity is a very important risk factor for the later life susceptibility and onset of gastrointestinal disorders. In particular, some of these disorders called functional GI disorders, functional bowel disorders. And this includes IBS, which is a highly prevalent gastrointestinal disease associated with stress and associated with early life adversity being a risk factor for disease development. And this is the most common diagnosis in gastroenterology office clinics. So about 40% of the diagnoses are from these conditions, such as irritable bowel syndrome, and a lot of them are stress related. And there's also other types of correlations between early life adverse events, whether it's injury, allergy, and other events that can serve as a risk factor. And this is the same for the animals that we study. And I'm going to be sharing some of the work that I've been doing, and this has really started as more of a clinical interest for me in veterinary medicine that has developed as we start to learn and understand how early life stress impacts animals in GI disease. And because we know that in animal production, there's a lot of stress that occurs during early life. And this, in turn, can lead to how susceptible an animal is going to be to certain diseases or disorders later in life. So it creates a nice similar paradigm and a model to study. And the general paradigm of early life adversity in GI disease that we're considering is that we know that there's a tremendous amount of gastrointestinal development occurring even in postnatal life. And the development is then going to set up this trajectory for lifelong gut function. However, if you have a stress during this time, this trajectory can be altered towards an increased disease trajectory. And this is what we're particularly interested in. What are these early life mechanisms here that happen in an adverse situation that are going to lead to long-term changes? And the model system that we've been using is looking at early weaning stress in pigs. And this is here a picture of a sow in a production environment with her pigs. Pigs are all happy. They're with their brothers and sisters and with their mom. And at a very early age, they're weaned from their mom. And they're put into a nursery environment, and they're mixed with unfamiliar pigs, a novel environment. So this is a traumatic stress on these animals. And even though the animals are well managed and well taken care of from welfare and health perspectives, there are just some inherent stressors, such as maternal separation, abrupt changes in environment, some social hierarchy changes, as well as just some aspects of the environment that create a significant stress on these animals early in life, and it generates a neuroendocrine stress response in these animals. But although it's known that some of these stressors can impact gastrointestinal disease susceptibility, the long-term impacts on their development and how they may be similar across species has not been studied in detail. So just a little more about our model that I'll present some data about. Again, it's centered upon inflicting or subjecting animals to just an early life adversity. And we know in nature the pig will naturally wean itself from the mom at about 3 months of age. And this is a time when most of the gastrointestinal tract has already been developed, both immunologically, functionally. And this is when they naturally begin to wean themselves. In an early weaning environment or early weaning stress, I'll denote this as EWS, you can see that this is occurring at a much earlier stage in gastrointestinal development. And we are interested in how this influences the long-term development. And our control that we've been using is a late weaned pig. So really just about a couple days later in development, where we can subject an animal to the same procedure and then look at how that may influence the long-term gut development. And this is just serum cortisol levels of pigs 24 hours after they've been weaned, whether they've been early weaned or late weaned. And as you can see here, whether the animal was weaned early or late, they still have elevations in cortisol and CRF. And I wanted to make this point because it shows you that no matter where in the response or where the animal is weaned, they're still exhibiting a significant stress response. So they're a very similar stress response. Now we're interested in how that different stress response at a different stage of life can lead to long-term gut function. We have been looking at different aspects of early weaned pig, and one of them is clinical aspects. And just to share some of this data on what happens in an early weaned pig, we've done a study where we've looked at some scoring, just some fecal scoring patterns in pigs that were early or late weaned, and then began to look at them throughout life and into adulthood. And as you can see here, this is just percentage of time pigs that are exhibiting this chronic sort of functional diarrheal disease in these animals, which is very different from what we see in the rodent models of early life adversity. They're not susceptible to these changes in bowel disorders or even diarrheal disease. And this is a typical pattern of what it looks like where an animal is weaned early. They just exhibit this relapsing loose stools throughout adulthood. And this was a study where we did a fecal scoring system, and it was actually according to the Bristol Stool Scale, which is a similar scoring system that's used in treating and understanding irritable bowel syndrome in humans. So this offers maybe another potential advantage of the pig, is that you can actually use some human scoring systems that are clinically used as well to assess what type of functional bowel disorder is there. And we also determined that this was functional because this is not an infectious pathogen-mediated diarrheal disease. There's no association with any type of common swine enteric pathogens. This is really just functional changes that have resulted in response to an early life stress. Interestingly, as we began to do more of these studies and repeat them, we began to look at differences between sexes in these animals. And this is the fecal scores and percent time spent with diarrhea. We can see that males and females both exhibit some patterns, altered functional diarrheal disease, but females are significantly exhibit more pathology and clinical disease. And this is important because a lot of the stress-related GI disorders in humans, there is a sex prevalence in these where, for example, IBS, women are 2 to 4 times more likely to exhibit more severe disease in the onset of irritable bowel syndrome. So this was very interesting that we're seeing more time with diarrhea as well as severity of diarrhea in females. And then we've also shown that these animals are more stress sensitive. When you expose them to mild stressors later in life, it's the animals that were early weaned or stressed that have more severe clinical diarrhea in response to that. Then we began to focus on some barrier function or some mechanisms, and I'll go through just a few features here. One of this barrier that is lining the gastrointestinal tract, this epithelium, and it's a little bit more about this and the significance of it. This is a cartoon of this epithelial barrier, which has many components, including microbiome, tight junctions, which essentially restrict luminal pathogens and toxins from getting across the gut, while at the same time allowing it to efficiently absorb nutrients. But then breakdown of this barrier, which is a central component of a lot of diseases in GI disorders, leads to this inflammatory response and a lot of neuronal sensitization, a lot of factors that can happen due to this leaky gut. And this is a common pathophysiology. So we were interested in how this disease affects that. And there's also some immunosuppressive properties too, if there's too much immune system activation. So we looked at long-term effects of early weaning on intestinal permeability in pigs. And what we showed was that in response to weaning, the early weaned pigs had this much more marked intestinal permeability dysfunction, but it not only is evident here, but as the animal ages, they still exhibit this persistent leaky gut in these animals, and this is into adulthood. So there are some permanent changes that are occurring just from exposure to an early life adversity. And this is measured by permeability on Ising chambers here. Again, we looked at potential sex differences in this response, and we also show this is permeability here. This is at 60 days post-weaning. This is 150 days post-weaning. As they are reaching adulthood, you can see that the males and females are exhibiting higher intestinal permeability or leaky gut. And then as you get into adulthood, females are the ones that still exhibit this persistent leaky intestinal barrier, which is very similar to human GI disease. And this is just demonstrating the importance of the barrier function in the gut and how it's linked to a number of important diseases. So this is an important model of potentially risk factors to later life disease. I talked about the enteric nervous system and the complexity between larger mammals and as well as compared to humans, compared to the rodents. So we began to look at that as well. We know that ENS is very important for maintenance of normal gut function, secretory absorption, regulation of inflammation, defense mechanisms. But we know that when this system becomes disrupted, there's a lot of disease susceptibility as well. And it's very plastic in the early life environment, which is relevance to early life adversity. But what we showed in this model was looking at early weaned pigs versus late weaned pigs. If you look at as the gut developed, this is the number of neurons stained by PGP staining. You see that there's a persistent elevation of the numbers of enteric neurons in the early weaning-stressed pigs. And we also assessed some functional aspects of the gut, looking at neural evoked secretory mechanisms, which really just target some of the main disruptions or disorders of functional bowel disorders here by stimulating the nervous system and measuring secretory activity in the gut. And this is the secretory response here. And you can see that early weaned pigs, due to graded doses of this neural agonist, have much more higher sensitivity to neural evoked secretion. So their enteric nervous system function has changed. Also, this is earlier and then this is later as they approach adulthood. And as we've shown before, again, the female animals are more susceptible to this early life stress-induced changes in the enteric nervous system as well. So we've shown increased enteric neuron numbers, hypersensitivity to neural evoked secretory mechanisms, which have implications for diarrheal disease, as well as some of the other functional aspects of GI disease. And also that, again, females in these are exhibiting a more heightened disease susceptibility. And then just a little bit more into this, what we've been characterizing is that we found that early weaning-stressed pigs are developing this cholinergic neural population that persists as they age, as opposed to the late-weaned animals which decline. And this is just a picture of some chat positive, which is a enzyme involved in acetylcholine synthesis, here in the submucosal plexus as well as the myenteric plexus. and looking at acetylcholine production elevated in the early weaned animals, as well as increased enteric cholinergic tone in these animals, which just suggests that there's some long-term changes in the enteric nervous system of these animals. As we began also to look histologically, we were surprised that there wasn't much of a difference between histologically between early weaned and late weaned animals, despite barrier disruption and diarrheal disease, histologically they look pretty similar, and this has been looked at by several veterinary pathologists blinded to these treatments, and just not remarkable. But one thing that we did find was this expansion of mast cells, which is an important innate immune cell in the gut that doesn't get picked up on normal H&E staining. And as you can see here, early life adversity or early weaning in the pig results in these elevated numbers and persistent elevation of intestinal mast cells here. This is the late weaned animal, this is the early weaned. You can see this is actually here, they're co-localized to enteric ganglia. So this neural immune communication in the pig. And what we found was that for sex differences, males and female animals both exhibited increased numbers of mast cells in their gut long term, but there was a difference with the females in that they released more mast cell mediators that are involved in mediating diarrheal disease, such as the tryptases in abdominal pain, in functional bowel disorders that are commonly seen in humans. And we've actually recently published a paper looking at the transcriptome differences between male and female mast cells and how they're translational across different models. And it kind of definitely fits this picture that there's some sex-specific differences as well in the mast cell populations. So in summary of this model, we think that this stress, whether it's an early weaning stress or another type of adversity in life, leads to this altered trajectory in gut function and development that's going to lead to a more increased susceptibility to a number of different disorders, especially in the gastrointestinal tract. And we've been characterizing some mechanisms here, including leaky gut, changes in the nervous system, upregulation of mast cell activity, as well as sex differences by using this model. And we've also published a number of papers in this model looking at some of the mechanisms targeting some corticotropin releasing factor pathways, as well as some susceptibility models to disease. So it really has become a model where we can study both the impact of these production practices on pig health and development, as well mirroring some aspects of human pathophysiology. And this can be shown here if you look at some of the things that we've learned from this early weaned pig that are similar to irritable bowel syndrome. We know that early life stressful events are a risk factor in human IBS, and this is an intense area of focus, trying to understand what in early life can change the susceptibility. We know that there's altered bowel habits associated with these conditions, both in the pig and human. Intestinal permeability is an essential pathophysiologic component of the disease. Enteric nervous dysfunction and hypersensitivity is also very similar. There's a sex prevalence that I mentioned before in stress-related GI disorders in women, with a higher prevalence there that we also see in pigs as well. And also the increased mast cells. And there's been a lot of interest in looking at potential therapies for targeting mast cells in human conditions such as irritable bowel syndrome and abdominal pain disorders, because these mast cells show up in higher numbers and they're all more closely localized to nerves, which fits the pathophysiology that we've been seeing. So to end, to summarize just some aspects of some translational implications of the pig as a model or dual use model here. I hope I've convinced you today that pigs are excellent models for gastrointestinal disorders, and in particular, some early life disorders that are emanating from early life influences due to their comparable anatomy and development, their clinical presentation of disease, and also their central nervous system, their development, which holds promise for beginning to study complex interactions between the gut and the brain, which are central to these diseases. We know that now, it was once a limitation, but genetic tools and reagents now are available in the pig models and porcine models. So there is a lot of promise to continue to be able to develop these models and study them even in a more mechanistic manner as well. There was also, if you think about housing considerations, which was part of the charge of questions that we've been asked to address, in general, pigs are can be housed in a general lab animal environment. It's not like their size, especially with the use of miniature swine available. They don't require highly specialized housing conditions, and they're pretty docile, very intelligent, very friendly species to work with. And also they can make an excellent model for preclinical models as well. So in addition to studying mechanism, can they be also a valuable model for testing out therapeutics? And this could be also a potential area of growth as we're trying to bridge this gap between potentially failed drug experiments that have been seen in rodents, but or it's been successful in rodents but failing in humans. This could be a potential model, and it has received a lot of interest from pharmaceutical companies as well. So again, I have a lot of people to thank that are involved in as collaborators and students that have been so important for driving this research forward, as well as the lab funding. And with that, I'd love to take any questions you may have.
Thank you, Sean. Anybody has any questions? Ask it.
So Adam, I was intrigued with the fact that you're seeing these changes that are at 60 days and 150 days. Reminiscent of some epigenetic changes, have you looked to see whether there are any changes in terms of that might be happening in these early weaning pigs?
Right. Interesting. Very interesting question. That's our hypothesis. One of our hypotheses is starting to look at some epigenetic changes. We have not entered that in the porcine model yet. We've developed some rodent models that begin to see some changes with that aspect, especially in the immune cells that are being developed. But we would love to also bring this into the large animal model as well.
You saw the effects later on. If you were to oophorectomize the pigs, what would you think would happen in terms of the effects of estrogen?
Very interesting question also. So what we see in our models is that we see a lot of this pathophysiology occurring well prior to sexual maturity in these animals. So it's 60 days of age, these animals are sexually immature from that aspect. So we are hypothesizing that a lot of this sex differences may be occurring even earlier, prior to adult sex hormones. Now, we do believe that adult sex hormones are probably playing a role potentially later on in the disease, but so that's something that we really do need to consider in this model. I should mention to you, as you brought this up, was our males in this study were male castrates. And so we were seeing differences with castrated males, which in that sense alone suggests that there are some significant potential sex differences there. Now we're comparing them to intact animals as well. So there could be some earlier sexual differentiation, genetic or perinatal hormonal influences that could be setting these animals up. And then also looking at them later in life, looking at some of the adult sex hormones. So very interesting, definitely interested in pursuing that.
Question over you. You looked a little bit at mast cells. I'm just wondering what type of immune function capabilities do you have in pig model? I'm naive to that. Relative to like human immunology or primate immunology, or developed in mice, can you take advantage of that, or is that still nascent? Or thank you.
There are a number of basic immunological approaches that you could use in the pig. So the development of porcine-specific antibodies for the immune system has been growing. You can do flow cytometry. You can do characterizing. A lot of ELISA kits are available now. So any major immune mediator or response, you can measure essentially the same as you would in a rodent tissue. So there are a number of tools that are out there, and we're characterizing some of these responses now with the mast cell now being a major player in this. Can we characterize the type of immune response that's developing, Th1, Th2, some T regulatory cells. So these are all pretty standard, but can be done and are already established in the pig.
Tried any dietary interventions or something during that window so you can prevent the progression?
Right. The question was, have we tried any dietary interventions early? And this comes from more of our work in agricultural related research that we do as well, which is we've looked at some feed ingredients, whether they're iron levels, which have been known to also high iron can be also very damaging to the gut, that can lead to long-term changes. But as far as really beginning to test out some preventative factors, we haven't. But it would be a really outstanding model to begin to intervene, especially in the area of whether it's formula, infant nutrition, or just some dietary strategies.
Congratulations, it's beautiful models. I have a question, and I'm from Michigan, from medical school over there. I see all of the good models you people present. How you are able to convince the people in other medical schools, the GI doctors, is going to use your model to advance the science? If you make impact, the money will be coming, right?
Right. Yeah, that's the difficult part of it, is these are excellent models, and they do have some advantages, but at the same time they may be disadvantages. In models like this, looking at the variability, I believe Dr. Hartman mentioned it before, a lot of these are outbred species, so they may better represent a human population. So looking at precision, opportunities to look at precision medicine, looking at specific populations is now possible. Some other obvious advantages would be from their size, just if you're using devices or making implants or other types of approaches that would be more amenable. That's also been discussed this morning. But it is a difficult, I'm not saying it's a difficult sell. I have no problem here saying that the pig is more advantageous to study GI disease than rodents, because I use both models, and we use both. It's just highlighting potentially the nature of the stressors that we're using here, and then the clinical outputs, and then how specifically the physiology that we're seeing can be more similar to a human or a larger animal model is the key, is to finding those select little key facts or unique physiology that may happen in these large animal models that are absent in the rodent. Just identifying those gaps, and then which is going to create just the importance or the translational significance of these models, helps support that.
Well, thank you, Adam, and let's thank our speaker.